Good morning. As Marie said, I'm Christina Jones. I am one of our technical project leads for some of our metabolomics efforts here at NIST. And I'm going to give you kind of a brief overview of NIST. Scott went into that, so I'll make it brief and kind of hurry it along. Our process for developing reference materials, which we are actually kind of evolving, and so I'll talk about that as well, and how we're trying to apply this new model for some of our omics efforts and materials that are in the pipeline. And then briefly discuss our plan, kind of like a high-level overview of our plan to develop a whole stool reference material. And you guys are here later this morning, our efforts in doing that and bringing that more to fruition. So we were created in 1901 as the National Bureau of Standards. We are one of the first, or were one of the first federal research laboratories established. We're also sometimes known as Industry's National Lab because of our mission, which is to really advance innovation and industrial competitiveness for the U.S. And so, as Scott mentioned, we are a National Metrology Institute, and we are that for the United States. And our kind of mission and what we do is actually established by the Constitution. I'll go through that. We're divided into 5 laboratories and 2 user facilities. And Scott mentioned again, for this work, it's our Chemical Sciences Division and our Biosciences Division, and we are in our Material Measurement Laboratory here at NIST. And so our measurement laboratories actually provide measurements that underpin the international system of weights and measures. We have 3 technology laboratories, and then there are 2 centers or the user facilities. And that's just to make sure that U.S. researchers across all sectors have access to specialized measurement capabilities. And so over the past few years, we have about 7 strategic priority areas, and one of those is the bioeconomy. And so that's really important for what we're at the workshop to discuss. NIST is committed to actually helping support measurement science research efforts in personalized medicine, metabolomics, the gut microbiome, or just microbial research, period. And we'd like to do that by providing tools and developing tools that help shift these areas from proof of concept and just like lab research studies to actually market realization and clinical translation. And so, we do this in different ways. We have standard reference data products. Oliver mentioned our NIST Mass Spectral Library, which is probably our most visible standard reference data product. It's really popular, particularly among gas chromatography, mass spectrometry users. We do calibration services. A great example of that is work that we do with Boeing to actually calibrate their force transducers so that they can ensure that their force measurements are traceable to the SI. And then we also have a variety of standard reference products that many of you may be familiar with. 80% of those standard reference products are actually chemical composition standards that are divided into 2 large categories. Our calibration standards, which includes high purity neat chemicals, as well as complex matrix standards such as food. There's a peanut butter, does this work? Peanut butter SRM that's pictured over on the left. And then our plasma, blood, as well as, this whole stool reference material would, fall under an extremely complex matrix standard category. So I wanted to pause and actually discuss a few definitions. On your agendas, I'm pretty sure the first page, there are some definitions related to NIST reference materials. And so a lot of you, the talk has been like SRM. So we make standard reference materials. We also make reference materials. At its core, reference material has to be homogeneous and stable. So, when you buy it at time point 0, it needs to be the same thing. If it's on our shelves and we're providing it to the public, you should essentially have the same thing when you measure it, or see the same thing, 10 years later, we're still providing it to you for you to purchase. And so, okay, it has to be homogeneous and stable, but then we also characterize it for one or more chemical or physical properties. We can actually take that up a level, and we also make certified reference materials. And so certified reference materials have certified values or specified values that are very close to the trueness of whatever that measurement is. So, if it's a metabolite or we're looking at cholesterol, then that means that if that's a certified reference material, the value for cholesterol is we're highly confident in that value. We've accounted for all sources of bias, and it's not the true result, but it's as close to it as you could probably get. And NIST makes materials that are known as standard reference materials. It is the same thing as the certified reference materials. All NMIs, all other NMIs, they sell their materials as certified reference materials. And this, we trademark standard reference materials, but it's the same thing. And again, our certified reference materials, or SRMs, can have two different categories of values associated with them. So the value is either certified or it's not certified. If it's certified, again, it's a very, very close approximation to the true value, and we've accounted for different sources of bias. So if you use one measurement technique to actually analyze and to obtain that value, another measurement technique will give you the same thing, within the uncertainty that we provide. We have non-certified values, and so those, you get a value, but that uncertainty that's associated with the certified value, that's not there for the non-certified value, and that's the biggest distinction. In the past, our non-certified values were known as reference values or informational values. Again, our process is evolving, and so now those are just kind of grouped together as non-certified values. We're not the only people that make certified reference materials. Industry makes them as well, but what distinguishes our materials from industry and other NMIs, what distinguishes NIST and other NMIs materials from industry is the fact that our certified reference materials are traceable to the SI. The other point I'd like to make is that our reference materials can also evolve into standard reference materials. So we may start off with just a reference material, but we can add certified values to that later and elevate a reference material up to a standard reference material. And so our traditional process that we've used over the years to develop our SRMs is actually pictured here. And so we typically have an SRM idea, and then we start designing it. Sometimes we'd have stakeholder input in the past, sometimes we didn't. But once we designed that SRM, then we work to actually acquire material. And again, we design SRMs, we want them to last. So when we acquire material, is usually a lot. And then from there, that material is packaged, it's processed, and we look at the homogeneity. So we want to make sure that, that packaging and processing, we have the same product over our entire production lot. So we define what the homogeneity would be for that material, and then we study it to make sure that the material is homogeneous. We characterize the material. Characterization is different based on the material and based on our perceived need or the customer's perceived needs. And then we look at the stability over time. Even when the product is released to customers, we're constantly checking that stability every 3 to 5 years. We document the entire process. We have information about the methods that are used for the characterization, and then it's released to the public. This process can take a really long time, though. It's usually about 5 years. It can be 7 or 8 years. And so one of the reasons why this process is evolving for us is because we want to cut down on that development time. The material characterization takes up the most time, and again, that's because for a standard reference material, it's traceable to the SI, and so we're using different methods to assess we're using different methods to actually assess and obtain those values, those certified reference values. We're making sure that we have calibrants throughout this entire process. Those calibrants are what are usually traceable to the SI. And then we also have other materials that we're using on top of those calibrants to validate everything that we're doing as well. So again, it's a lengthy process. And as an example, many of our metabolomics stakeholders are familiar with SRM 1950. Oliver mentioned this as well. And so it's metabolites and frozen human plasma. It was designed for method validation of specific metabolites in normal plasma. And so it's certified for over 40 individual metabolites. I think it's closer to 45. And we also have about 45 or a little more than 45 non-certified values for metabolites. One vial or one SRM unit contains 5 1 milliliter vials. So you're getting 5 mils The cost of that is $1,100, but it's important to point out that, you can buy plasma from anywhere, right? You can buy plasma from anywhere to use as a QC sample with your studies. What you're paying for is those values, and those values are listed on a certificate or included on a certificate of analysis that comes with our materials. But what's interesting is that while there are people that are using this material for the values on the certificate, like the clinical chemistry community, the untargeted metabolomics community really uses this material as well, and it's an off-label usage of the material. So they're not necessarily just looking at those concentration values that's listed on the certificate. They're using it for things such as, evaluating instrument stability over time or to develop methods and things of that nature. So that begs the question, should we only be making certified reference materials? In the past, we've really focused on our SRMs, but should we focus more on our reference materials now? And so, again, a certified reference material is used to establish SI traceability. It's used to support or help industry support regulations that their regulatory requirements, and then also establishing trueness of results with extremely high confidence. A reference material, though, can be used to assess variability. It can be used for method development. It can be used to look at your instrument stability over time, as well as comparing results among labs. So again, if you're not establishing SI traceability or at a certain point concerned with, having a very high confident value, then a reference material could suffice. And, based on this is not just SRM 1950, but some of the off-label uses of our other SRMs, it just led us to rethink our process and to start engaging with our stakeholders a lot more. And so now what we're starting to do is, work with our stakeholders. We may have an idea for a reference material, or stakeholders might have it, but work with them to prioritize our reference material development, as well as identifying how that material should be designed, working with them to screen the materials. So now we're doing a lot of interlaboratory comparison exercises, or even just working one-on-one with different collaborators to screen the materials while we're screening them as well. We're developing quality assurance programs, which is essentially just a formalized way of doing interlabs, where we help people or different scientific communities understand where their measurement variance is, also helping them to get better at measuring, let's say, a specific measuring that they may want to, and then gathering, compiling all of this information and making a decision. Does this material need to be an SRM? Should it be an RIM? Do we want to get it out quickly and have it as a reference material, an RIM, and then turn it into an SRM? Or do we just want to keep doing formalized interlabs with this specific community? So we acquire material and just send it and have them constantly evaluate it and just better their measurement methods. And so this is the model that we've been using with the untargeted metabolomics community in developing our new materials. One thing, and many of you know, is that, analytical data that you get from your metabolomics research can sometimes be more variable than actual biological changes that you're investigating. Metabolomics workflows are extremely extensive. Every single step introduces technical variance. And so how do you evaluate that and get a handle on that? Also, we know that there's a lack of a large kind of commercial supply of QC materials that could be available, or it's that same material that's available over a long time span. We've been told that suites of materials are needed. So, metabolomics communities are doing differential studies. And so it'll be nice to have a suite of materials that you can take through that entire workflow that you always get the same answer for. And so, again, the new model, the biggest kind of thing is stakeholder engagement. What do you need? How can the material help you so that we better design it? And we started doing that a lot with the metabolomics community. On top of that, we also do metabolomics research. We have our researchers NIST researchers in Charleston that do a lot of metabolomics research, as well as some of the stuff in Gaithersburg. And far and large, what we've been told is that, we don't want quantitative values just yet. We want just qualitative information. Can you release a material that has metabolite identities associated with it or annotations, and then the confidence level with those? If it's a suite of materials, can we just have fold changes? So, I know that back to cholesterol, this is, the fold change or the relative amount between a certain group of materials, and then that can give me confidence if I'm measuring that same thing within different groups, different phenotypes of samples, phenotypic samples. The other thing was, well, then, what types of materials? And we kept hearing, okay, blood and urine, and then tissue, but blood and urine first as a priority. And so we took all of this information, and we've now established a Metabolomics Quality Assurance and Quality Control Materials Program. And the idea is that, again, deep stakeholder engagement, but we'll start developing materials that we release out and have the community analyze them through interlaboratory comparisons. And again, it's just qualitative information. Metabolite identities and annotations, as well as those fold changes, and then metabolites that help discriminate between a suite of samples. The interlab comparisons are also important. It helps us in the material assessment, but one thing about the omics field is that everyone has their own workflows and their own methodology. And because of that, we don't have access to all of that in our organization. So by utilizing people in the community, then we can get data that spans all of the workflows, the different type of instrumentation setups, and make that data publicly available to everyone. So if you see someone that uses the same type of instrumentation as you could compare your results to them. And so one of the things that we have in the pipeline, based on this engagement and feedback, is they call it a candidate RM 8231. It's not an RM just yet because it hasn't been released and we're still characterizing it, but it's a frozen human plasma suite, and this is specifically for metabolomics. And we've already in-housed in some NMR and LC-MS analyses where we see that these pools have different metabolite profiles. We mentioned that, the material has to be homogeneous and stable to be a reference material. And so one of the things we're starting to test for assessing stability is point-of-care analyzers. They're available for plasma as well as urine, but it's a quick, rapid assay, and it will be just easier instead of using, GCMS, LCMS, to use that to track, are our values still looking the same over the time? Is this material changing or is it the same? And we're also collaborating with our NIST Mass Spec Data Center, who produces the NIST Mass Spectral Library, to help us go through and annotate and identify what we're seeing in these materials. And so we started doing some interlaboratory comparison exercises. We have our own that's going to launch soon, but we've been working with SciEx and Leiden University to evaluate the lipidizer and what type of data do what type of data do researchers get when they use those materials with that instrumental platform. We just started working with the University of Florida and Avanti to kind of spike different things into the samples and see how that impacts analyses. We're working with the Metabolomics Quality Assurance and Quality Control Consortium so that its participants can also help us evaluate the metabolite profiles of these materials. And so in addition to the plasma, we have other things in the pipeline. We have a frozen human urine suite. We also have a human liver extract. And this one is different because it can be used to actually assess your system. It's a system suitability material. So you can use it to track, does my instrumentation, am I getting the same answer that I got 3 months ago? And this is, Tracy, this is still available for people to actually get. So if you want or you're interested in that, please talk to Tracy throughout the day because there's a kind of a pre-release launch going on with this material now. And then we have a human liver suite that's in the pipeline as well. And so we'd like to also extend this model to the gut microbiome research community. You guys know I'm kind of reiterating some stuff that Johanna has already said today, but there's been a tenfold increase in microbiome-related publications since 2010, and the trend seems like it's likely to continue. The NIH has invested a lot of money into human microbiome research that's separate from their investment in the Human Microbiome Project. There are products that's on the market where people can buy and actually see what their microbiome is, and then there are, kind of like health recommendations based on that. But the question is, are we at the point to where this research should actually be translated into a consumer product? There are a lot of research challenges, some that are, similar to just metabolomics in general. There's a huge body of work and ongoing studies, but there's not standardized approaches. It's still not at a point where labs can actually compare their data. And it doesn't help that the analytical matrix for whole stool is extremely complex. So, it is a field that's in need of reference materials to kind of help get a handle on some of these challenges. And so NIST has been working with ILSI as well as the Biocollective to start to develop a human whole stool reference material. Again, because it's a reference material, we have to ensure that it's stable and homogeneous over space and time. We'd like this to be produced rapidly and cheaply. And the intended purpose is just as a start for harmonization so that labs can compare their data, and then you can also use it for your other QA, QC purposes. But, not necessarily for SI traceability, not that right now there's a particular metabolite that you need extremely accurate concentration values for just yet. And this material will be used for metabolomics, DNA quantification, metagenomics, just a host of things. That's the idea. Or it can be a suite of materials, a suite of human whole stool reference materials that can be used for these different purposes. Our idea, and again, we're here to discuss this more today and to get feedback on this, is again that this is something, once we know how the preservation methods impact the stool, once we know that we have something that's stable and homogeneous, it can be released as just a first step to get a material out to the community. Then we can still characterize this in layers. We can go back and start identifying metabolites that the community wants values for, just identities for. But we can release it, get it out, and then do that over time. I forgot about this slide. And so then, same thing, what I just said. So first, most important thing is to assess for homogeneity and stability. We can get it out, do some interlabs, and then after that start to identify confidently metabolites that the community wants identified, which is partly what we're going to do here today at the workshop. And then from there, some of those will end up being extremely important. Some of them over time, there may be better evidence of the health implications. Then we can start assigning concentration values to those metabolites and evolve that material into a standard reference material. And so, again, our material development process is evolving, and one of the main things that's changing is we want stakeholder input. We want feedback. We're valuing that. It's helping us prioritize our efforts. We've had a huge focus on standard reference materials before in the past. We now are putting a larger focus on just our reference materials, because for emerging scientific communities, they can be used to assess your measurement variability, they can be used to develop protocols and analytical methods, and they can be used to harmonize measurements between labs, which is key if we want this to translate into a clinical setting. NIST, we're committed to making a human whole stool reference material, again with ILSI as well as the Biocollective, and that can eventually become a standard reference material. But things that we want to know is this a suitable plan? And I think that that's one of the discussion questions today during the breakout sessions. And again, one of the objectives of the workshop is what are key metabolites that we can identify in the future that will help propel your research to the next level. Another thing, and there are a few with the NIST researchers and people, please kind of like raise your hand or stand up. So you're identified. It's quite a few of us attending the workshop. One of the other things that we want to know is how do you actually plan to use a reference material? If you have a human whole stool reference material, how are you going to use it? And that's not spelled out exactly during the breakout sessions, but please talk about it during the breakout sessions, and we're here, so you can find us throughout the day and even have one-off discussions with us. Yeah, we'd like to know, like, how will you actually use the reference material, or any other information that you want to provide to us. And I just want to acknowledge everyone that's working on our materials on the pipeline, as well as the NIST poop team. All right.